So the last portion of our overall metabolism is going to be electron transport and oxidative phosphorylation. So I just kind of breezed over in the last sections talking about that we make NADH, we make FADH2 and everything, but I just said those make ATP. I didn't actually say how it happens. So electron transport and oxidative phosphorylation is how it happens. So just as a review, from 1 unit of glucose so far, from 1 unit of glucose in these processes, we make in glycolysis, we make 2 ATP and reduced coenzymes, so we have 2 NADH. From the oxidation of 2 pyruvate, we make 2 NADH, so that's getting 2 pyruvate to this acetyl-CoA. And then citric acid cycle with 2 acetyl-CoAs, so remember, we go through it twice just because it's one glucose gets broken down into 2 pieces. We get 2 ATP, 6 NADH, and 2 FADH2s. So for 1 molecule of glucose, at the end of all of those cycles we've talked about so far, we have 4 ATP, 10 NADH, and 2 FADH2s. So the third stage, the last part of the third stage of metabolism, which is electron transport, and in this, NADH and FADH2s from glycolysis get oxidized and reduced, oxidized to provide energy for ATP, the synthesis of ATP. So these are going to be transferred into ATP overall. So electron transport occurs in the mitochondria. So this is, if you remember, the mitochondria is kind of like a bulb and it has lots of like membranes inside. It's kind of like, it kind of looks like a brain chopped open, right? It has all these little membranes inside, and these membranes look like this. So this, here's the inner mitochondrial membrane. Those are those membranes inside. We have the mitochondrial matrix here, and then intermembrane space here. So there's kind of like cavities inside. So this is overall the image for it. There's a lot going on here. So we're going to talk about it more in basics and general terms. So the chemoosmotic model, what this says is that protons from complexes 1, 3, and 4, so proton complexes, these 4 blue, those are protein enzyme complexes that have activity. So I just kind of tried to highlight the numbers there for you to see, but number 1, 3, and 4 move protons. So we have protons on, remember, NADH. So we move hydrogens, protonated or H pluses, from the mitochondrial matrix, NADH or FADH2. They move those protons to the inner membrane space. That creates a hydrogen gradient across the mitochondrial membrane. So one side is hydrogen deficient, one side is hydrogen rich. In order to get the protons back, we are going to do protons return to matrix through an ATP synthase. So that's this big guy over here. It's a enzyme. I think it's technically 4 different enzymes all together in a complex. And that is how, so the hydrogens go from the membrane mitochondrial space into the intermembrane space, and then through a hydrogen channel in the ATP synthase, that energy from the positive charge moving that gradient is going to be what powers the ATP synthase, so the enzyme that actually makes ATP. So ATP synthase has subunits that rotate as hydrogens move along the gradient. So it kind of just spins kind of like a turbine, think of it like that, creating energy. In those conformational changes, as the hydrogen goes through, it makes conformational changes that allows the binding pocket to turn into the active site, and it catalyzes ATP. So you go from ADP plus a phosphate to ATP because of the hydrogen gradient. Okay, so these reactions, specifically what happens in each enzyme complex. So enzyme complex 1, NADH plus H plus, this is a coenzyme. We're just going to call it Q. It's a coenzyme that's in the mitochondria and it stays there, but NADH plus H plus coenzyme Q makes NAD plus QH2. So really we're just moving those two hydrogens over to a different coenzyme, reforming NAD plus, just kind of stays on that side, and then it can go back and do more of those processes like the citric acid cycle or glycolysis. Enzyme complex 2 takes FAD H2 plus a coenzyme to make FAD plus that Q coenzyme H2 again. Enzyme complex 3 takes those QH2s that we formed from complexes 1 and 2 plus 2 cytosol Cs. Don't worry about what those are, but they're oxidized, and it releases that coenzyme back plus 2H plus into the intermembrane space and reduces those. We're not going to worry about the cytosol Cs. And then the last one is enzyme complex 4. Takes 4 electrons and 4 protons and 2 of those coenzyme Qs, and it forms water back down here. Okay, so that's what those reactions are. So what is oxidative phosphorylation? So this is how protons are involved in providing energy needed to produce ATP. It couples the energy from electron transport to the synthesis of ATP from ADP. So overall, what you need to know is that this is the overall reaction for oxidative phosphorylation. You take ADP and PI, phosphate, so ADP and a phosphate, energy from the proton gradient that you formed in those enzyme complexes. Using ATP synthase, an enzyme, you form ATP. I think, we'll do this. So oxidative phosphorylation, let's see just overall what we form in this process. So energy released from oxidation of NADH plus H plus, so 1 NADH plus H plus overall makes 2.5 ATP molecules. So you see the full balanced equation here. I'm not going to go into it too much, but long story short, NADH plus H plus makes 2.5 ATP. And then down here, FADH2 makes 1.5 ATP molecules. So you can see how much ATP is formed based off of the energy in these different coenzymes. Okay, so I think that's where we're going to stop this video, and we'll talk more about overall ATP formation in our next video.